Thank you. Thank you for the invitation. It's good to be here back at the Mesa and see so many friends here. So I wanted to talk a little bit about TreeFrog Therapeutics today. So my name is Peter Anderson. I'm the Chief Research and Development Officer at TreeFrog Therapeutics. We're a company that is headquartered in Bordeaux, France, and we also have activities in Boston and in Japan. So our motto is cell therapy for all. And what we mean by that is we are building a platform to industrialize cell therapy manufacturing. This platform we call C-STEM. For that, we focus on some major pillars that we believe are a key challenge in the industry. Cell quality, scale up, and clinical readiness. So what we focus on is really improving the cell health. So we do that by, and I'm going to show you that, by reducing external stressors from the system so that this can be passaging or shear stress, so you get a better quality of cells. We are able to minimize the occurrence of cellular abnormalities and thereby maximize viability and genomic stability, which we believe is a key issue. To do that, we obviously do that to secure the highest cell quality of our iPS-derived products. So when it comes to the second pillar, scale-up, we think on a commercial level. So when we do a program, or when we think about programs, we think about the very end, about the commercialization and the late-stage clinical trials. For that, we have a big focus on the scaling. So we don't just think about millions to billions. We're actually thinking about billions to trillions of cells. So our goal is to be able to develop batches of trillions, in a trillions number, for these therapies. With the C-STEM platform, we can maximize the predictability. We have a big focus on reducing the developmental and development and production cost, which we believe is a key challenge in the industry. So of course, we're making a robust and reliable system to meet the clinical and commercial needs. And if we go to the last pillar, the clinic access, what we're really trying to achieve, we've already demonstrated with our tissue format that we have greater efficiency, and I'm going to show you that with some data. We can improve engraftment and the viability with our product. But we also have a very high viability of the tissue that we generate. That means that we have a minimal loss upon transplantation. And obviously, to do all that, we need to reduce the developmental cost to actually get closer to the patient scenario so we can actually develop some affordable therapies. So this is C-STEM. C-STEM is an encapsulation technology. It's an alginate shell that protects the cells inside it. So inside this alginate cell, we can either use it as is, or we can define the matrix that we want to put in. So we can basically engineer microenvironments. And this is the key when you're working with iPSCs, right? So by mimicking the environment, we can mimic any environment that we believe is necessary for this potential therapy, or if it's just amplification of iPSCs. So, as you can see here up on the video in the top right corner, that's what it looks like when we culture iPSCs inside the capsule. So as you can see, the cells, they will form a rosette structure similar to an epiblast, and they will rapidly grow. So this way we can reach amplification rates of up to 300. But more importantly, what happens in there when we have them inside these capsules is that we're removing all the external stressors, as I mentioned before. That means that we don't have an artificial selection pressure on these cells. If you grow cells on 2D, they will compete for space. We don't have that here. And if you grow cells in a bioreactor, you have external stressors in form of shear stress. So we're eliminating that using this technology. So as you can see, here in the middle, let me see if I have. So here in the middle here, this is our encapsulation technology. So we're able to encapsulate iPSCs and generate thousands of these capsules with iPSCs inside. So from here, we can either amplify in bioreactors, we can amplify cells to high numbers, and we're reaching amplification rates of up to 300. or we can use the system to basically differentiate. I'm sorry, there's no more juice in this one. So we can differentiate into a final cell product, as we can see here. So we basically generate these 3D format products, but we can also dissociate the final product if we want. And when the cells then are encapsulated, we can remove the alginate shell by simply adjusting the calcium levels. So as you see in the video on the bottom. So this is the Sakura. This is a fully closed system that we have built at TreeFrog. It has a single set use set cassettes, and it's basically built for GMP setting. So we use that to basically encapsulate cells. So with that, we're achieving the scale and the commercial scale readiness, as I mentioned before. We have a very high consistency with this method, so we can reliable and take these encapsulated cells, put them in any type of bioreactor, and thereby we can amplify or differentiate cells. And what's really unique about this system that we've proven is that it's bioreactor agnostic. So meaning, because when we can take these cells, we can take them into small bioreactors, we can take them into large bioreactors, but because we don't need to worry about the shear stress or the physical properties of a bioreactor, we can eliminate that, and thereby we have a linear scale between bioreactor settings. So, as I mentioned, because we can use this technology to develop 3D microtissue, we've demonstrated that we have enhanced engraftment and functionality of the tissue, and finally, that the cells that we grow inside these capsules has a very, very high quality. We have, in this case, here for iPSCs, pluripotency is about 98%, with viability around 97%. So literally, because we're reducing the artificial pressure, we don't have the issue with cell death during the processing. So this is what it looks like when we use the Sakura. Because now, as you can see, we're industrializing the entire scheme. Because every single capsule that we produce here will have cells in them that forms the rosette structures that we can either amplify or we can differentiate. So when we can use this Sakura to generate thousands of these capsules per second, it means that we can rapidly scale up to the therapy that we want. So we currently have a pipeline where we focus on that is part license agreement, because we are open to licensing out our technology. So the first that you list here is an out license agreement we have with Vertex Pharmaceuticals that use our platform for the T1D program. And then we have our internal pipeline programs, Parkinson, and one for liver failure. The most advanced we have internally is our Parkinson program. So, touching on the Vertex partnership with Vertex Pharmaceuticals, that's our partnership that we initiated early last year. And here we're really working with them to scale to the needs that Vertex need for the T1D program. And this is where it becomes very important to think in trillions instead of billions. So with Vertex, we have like some high goals, and we set the bar high. So that's why we are really ramping up production with Vertex, so the partnership is it's going great. It's collaboration partnership, and we're making a good advance in that program. So here we have our images and some data from our Parkinson product. As I mentioned, it's a 3D format of dopaminergic cells. What we've demonstrated is that it can be cryopreserved, we can control dosing, and we have excellent preclinical time to effect and efficacy, as you can see down here on this graph. So within about 3 months, we see a good effect in the rat model that we use. We also have some very promising reinnovation data. We believe that reinnovation is one of the keys for Parkinson to make sure that your product really integrate and function well. So we're currently in the process of mapping out all that data and doing all the quantification, But some of the data here that I'm showing up here for the rat, we can see that we basically have protrusion from the explant of more than 5 millimeter. And when we look in our studies in primates, we see the same similar kind of cell protrusion from the graft. So we really see that once we have engrafted, we see the cells really protruding out and connecting with the rest of the tissue, which we believe is a very important key for the product. Finally, we have our liver program. It's an earlier program where we're generating hepatic microtissue with our C-stem technology. And here, as you can imagine, liver is the biggest organ that we have. It's about estimated to be 1.6 to 1.8 kilos. So we need to generate a lot of cells or a lot of microtissue to actually do an impact there. So here again, I want to emphasize the scaling that we're working at. We're scaling to meet these medical needs, and here we're talking trillions of cells and microtissue. But if we look a little bit more on the functionality of our microtissue, what we're pretty excited about is that when we compare it to other processes, in this case here as in 2D, we see that there's a lot of the functionality properties of the liver microtissue that competes the 2D production. So for example, when we look at albumin secretion and CYP3A4 activity and urea production, we can see that we have a much higher functionality of this tissue in general. So we're very excited about these results and about the product that we are able to make with C-stem. So on my final slide here, as I explained to you, we basically are working with two ways. We have two strategies in TreeFrog. The one is a proprietary or co-development pipeline. This is where we have our Parkinson product and our liver product, which we are always open to discuss and potentially discuss co-development opportunities around. We're also open for co-development or licensing options for the technology in general. And then we have our out-licensing model, as I mentioned with Vertex, where they have done an out-license of our C-stem technology to build their program and their needs for that. So with that, I'd like to say thank you, and you're welcome to come and talk to me if you have interest in TreeFrog or in the C-stem platform in general. Thank you.